So let's just do like a quick overview and slash tiny bit of details and like different things in the kidney, and then we'll move on to like digestion. Okay. So we'll try to do the kidney for the next half an hour. Ask any questions, like just interrupt me. Yep. All right. So let's zoom in. So nephron. Okay, cool. So blood is coming in through the afferent arteriole into the glomerulus where it gets filtered into the Bowman's capsule. That's fine, right? Now, what gets filtered? Anything that's small, which is water, amino acid, glucose, salt, but not like plasma proteins. So I wanted to kind of go into a little bit of the filtration part. So one of the things to kind of understand why filtration is more in the kidney, like in the glomerulus compared to let's say other places, because you also have filtration in like capillaries, other places as well, which also kind of we could talk about it a little because you have spoken about it previously. But it'd be great when you're answering questions to always talk about like hydrostatic pressure versus like osmotic pressure, oncotic pressure, and describe in those like using those terms. So for example, in the arterial end, you'll have a higher hydrostatic pressure in the capillary, which is why more filtration occurs, right? And fluid kind of goes out in the ECF. And then on the venous end, what happens is you have a higher osmotic pressure because you filtered all the small stuff, but didn't filter out plasma proteins like albumin, which kind of increase the osmotic pressure and cause fluid to go back up into the capillary, like most of it, right? Those are all familiar things that happens in the capillary. Okay, cool. So whenever you're talking about, if you're talking about these kind of things like, oh, what causes edema, talk in these type of terms, like talk about hydrostatic pressure, talk about osmotic pressure. So for example, if I say that, okay, what happens if the plasma protein in your entire blood like decreases? So how would that affect, or what would that cause, or how would that cause edema or something? So you'd be thinking about, okay, that's because it's the osmotic pressure that was bringing all these fluid back up to the capillaries and into the blood vessels. But because there are less plasma proteins, that osmotic pressure is less, which is why fluid stays in the ECF and outside, causing edema or something. Okay, so just wanted to bring that back in case you get any questions and how to like answer that. Okay. So you have more filtration in the glomerulus versus like other places because of three main reasons that I like to think about. One is the efferent, which is like the one that's leaving. The efferent is actually narrower than the afferent, which is why pressure builds up in the glomerulus. Is that okay? Right. Also, the glomerulus is like very tortuous and twisty, which kind of causes pressure to build up as well. Another thing to think about is just this is an arteriole, which means it's connected to the artery, which is connected to the heart, pumping blood. So just normally it would have like higher hydrostatic pressure as well. So just those things. Okay, great. Is that okay till now? Okay. Then we also talked about what is glomerular filtration rate, right? That's just like, okay, how much fluid's getting filtered per unit time, right? And that depends on like generally two things. One is net filtration, the stuff we have talked about, stuff to do with pressure. Another thing you guys have talked about as well, like KF, which depends on like surface area and permeability and everything. That kind of ties into thinking about podocytes. Does that sound familiar at all? Yeah. So podocytes, they're like a barrier, doesn't allow big stuff like plasma proteins to get in. So another cute question used to get, if you get plasma proteins in the urine, how could that be like tied in, or what could be a possible issue? And one of the possible issues like damaged podocytes and allowing plasma proteins to like get into the urine. So yeah, anyway. Cool. Yeah.
In terms of like stuff, like there were like
Yeah, we're gonna come to that.
oh,
Okay. Right. Yeah. KF is coefficient of filtration, right? Yeah. They do, but sometimes like I'm saying like it doesn't allow all the plasma protein, but usually it's supposed to block all of it. Like no plasma protein is supposed to be in the urine, ideally. So if you find some, that kind of is indicative of like, okay, something may be wrong in the filtration system that it's allowing things to filter out. Yeah, but most likely it's like they talk about like podocytes. Because another thing is podocyte barrier is dynamic. So it does have the ability to become tighter or looser based on what's needed in terms of rate of filtration. Okay. All right. So moving on. So things are filtered, cool. They're going to proximal convoluted tubule. What happens in the proximal convoluted tubule? Reabsorption of what? Most of the glucose, all the glucose, some of the glucose, all of it. Okay, all the glucose are reabsorbed. What else? Most of the salt is reabsorbed, I guess. Like 2/3 of salt is reabsorbed, 2/3 of water is reabsorbed, amino acids are reabsorbed. So like everything, most of the reabsorption happens here. Great. And we can kind of go into details of like different reabsorptions and how does that happen, which is kind of like did talk about in the lecture. So how is sodium reabsorbed? They have like sodium transporters because you have the ATPase sodium potassium pumping sodium, just like labeled B as like the basolateral side, A as the apical side. But yeah, so the ATPase would pump sodium back into the blood on the basolateral side. That would create the sodium concentration gradient, allowing sodium to be reabsorbed from the lumen of the tubule into this. Okay. Is that okay? And if sodium is reabsorbed, chloride follows because charge, and also water follows. Okay. Next, let's talk about the sodium-dependent secondary active transport, which is the way glucose is reabsorbed. Yeah. If you recall, we're using the concentration gradient of sodium to kind of get in glucose as well. Is this okay? Yes, no, nodding. Okay, cool, perfect. Great. Does this look familiar? Kind of talked about it in the group problems as well, quite a bit about reabsorbing glucose and how it could be like maxed out because they're using transporters. So beyond that, if it's above that level, like the TM, right? Above that level, then it's gonna be showing up in your urine. Yeah. Okay. So that's okay, right? Next one. We also talked about organic anion secretion, which is like the tertiary active transport. Right. Let's talk about that a little bit. Very clumsy. And I was gonna draw all of this live, but I was like, I'm gonna mess things up. So let's start from the beginning. So you have the sodium potassium pump normally set up on the basolateral side. That is fine. So it's allowing sodium to get out of the cell. Okay, all right. That causes sodium to build up outside here, right? Which is why sodium would want to go back in. So they're gonna use this sodium dicarboxylate cotransporter to go back into the cell. That's happening here, this purple one. So when sodium is going back into the cell, they are dragging alpha ketoglutarate with it because the cotransporter. Okay, that's fine, right? Okay. So because there's more alpha ketoglutarate just building up inside the cell, they would want to go out, right? So they want to go back out using the OAT, the organic anion transporter, right? So they would want to use that to go out. That's this part. And while they're going out, they actually have to pump in or actually get in just organic anion into the cell. That's fine, right? Okay. And now because you have organic anion into the cell, they would want to, like, if it's built up here, they would want to go out. So they're going to use the O channel on the other side to get out. Is that okay? And that kind of like talks about how you're secreting these organic anions, for example, by salt, benzoate, saccharin, all that stuff, out into the lumen to be excreted. Is that okay? Yeah? Okay. So we talked about filtration, we talked about some reabsorption, we talked about some secretion. Right. Okay, great. Yeah. Yeah. Mm-hmm. Yeah. Yeah, exactly. So you also have that on the apical side, like you also have the sodium dicarboxylate co-transporter on the apical side, just building up that alpha ketoglutarate concentration. Is that okay? All right. So like I said, we talked about filtration in the glomerulus, we talked about reabsorption here, we talked about secretion here. So those are the three things that happens. And if you want to think about what gets excreted at the end, you would have to think about all the three processes that are happening. So let's talk a little bit about, like, at the end, what is getting secreted or excreted, sorry, would be whatever is filtered. So all the small stuff, water that was filtered, minus anything that was reabsorbed. So like the sodium was reabsorbed, and glucose was reabsorbed, anything that was reabsorbed, plus anything that was secreted, like directly into the tubule. Right. So those three processes. Also, I'm actually gonna post all of it. So, okay, all right. Cool.
What's the difference between like tubular secretion versus
So filtration is just like a passive process. It's happening here, just under pressure. So it's a passive process under pressure. Whereas secretion is more like an active process. They're actively getting some substances directly into the tubule, and that's happening.
It's like very it's not specific at all. It's very general.
Yeah. Okay. Some drugs are also like secreted out using that. Okay. All right. Coming back to clearance. So I guess you did some math in the group problems about clearance. So you kind of can calculate how much a particular compound would be cleared out of the blood to go into the urine. So, and this is the equation you use. You divide the excretion rate by the concentration of that compound in the plasma to kind of find out the clearance rate, which is like how much of like particular substance dissolved in per ml of plasma would be excreted, like with respect to time. So, why is that important? Two things. One, that's important for like doctors for things like deciding, okay, how much drug to give, because drug concentration depends on how much it's binding versus how much it's getting cleared out. So they need to know that if this drug is like very easy to like get out of the system or that it's safe for a longer period of time. So that. Second, we could also find out how it is like interacting with the kidney. So for example, is it getting reabsorbed? Is it getting secreted, filtered only, or something, right? So if you have a clearance rate that is lower than your GFR, what does that mean about that drug? GFR is just like filtering everything, right? And instead clearance being less than GFR, which means what? Yeah, it's reabsorbed. What if the clearance rate is higher than GFR? Secreted. Is that okay? Yeah, okay. And what if it's equal to GFR? Just filtered, right? Is that okay? Cool. All right. So moving on our journey. So the liquid in the tubule, they continue along the loop of Henle. The descending part only reabsorbs water. The ascending part doesn't reabsorb water because that part is impermeable to water. They just reabsorb like salt, like sodium, chloride, potassium. Is that okay? Yep. This one? The active, right? Yeah. All right. Cool. I wouldn't talk much about the gradient that is being set up just because you guys built the gradient during a class activity. So I'll skip that. But let's talk about what happens in distal and convoluted tubule. So what happens in distal convoluted tubule? Reabsorption of something. Yeah. Salt. Yeah. So you're getting reabsorption of sodium generally. So distal convoluted tubule and collecting duct, they both reabsorb salt and like water, like respectively. But you could actually fine tune this part. So up till like proximal tubule, we saw they were just reabsorbing or secreting and stuff, and they were doing their own thing. But over here now, the brain and nervous system and everything can manipulate it a little bit more and can fine tune it. So depending on like what hormones are being released, right? So let's talk about the reabsorption of sodium and what hormone is that dependent on in this case, like in the distal convoluted tubule. It's right there. I don't know why I'm asking. It's aldosterone, right? So yeah, so let's focus on this a little bit more. I'll delve into deeper in this part, but basically if you have more aldosterone, right, you will that aldosterone, like it's a hormone, right? And if you think about it, it's is it a peptide or like steroid hormone? Yeah. So a quick tip, anything that is like a steroid that kind of has that in its name, aldosterone has steroid in its name, so that kind of gives you a clue that it might be like a steroid hormone. So like corticosteroid, all this stuff does have like that in the name. So that's a clue. So aldosterone is a steroid hormone, which means they have a receptor to bind, or they could like directly go into the cell. Yeah, and go into a cell, their receptor is inside. So they would change transcription and translation of what? I guess you talked about like two things. One is the ENaC channels, right? Which is important. So there will be more ENaC channels placed on the apical side so that more sodium can like be reabsorbed. Is that okay? Okay. And they also put more sodium, potassium, and ATPase on the basal lateral side so that more sodium can like go into the blood. Is that okay? Cool. So that's aldosterone. And we could talk about what increases aldosterone. I mean, you know that already, which is increase in renin when the pressure falls, when blood pressure falls, right? Other things, for example, increase in potassium also does increase aldosterone release, and increase in osmolarity. Oh, sorry, increased osmolarity actually inhibits it, so, yeah. Which kind of makes sense. You don't want salt to be reabsorbed if you already have a high osmolarity. Is this okay? Don't know. All right. Okay, I need to probably rush a bit more. Okay. Sure?
Yeah.
Okay. Okay. Cool. All right, let's talk about the collecting duct and what happens there. So you have reabsorption of water in the collecting duct, right? And what influences that? ADH? Right. Okay. IDH or vasopressin. What do Americans say? Vasopressin? Okay. Okay. Sorry. I think I wrote ADH everywhere. So vasopressin. Okay. All right. So if you have low blood pressure or low blood volume or high osmolarity, all of those would trigger release of vasopressin, right? So if you follow, baroreceptors detect low blood pressure, sends impulses to like hypothalamus. I'll also upload the whole document, so don't worry about it. Okay. Yeah. So baroreceptors would detect decrease in blood pressure, would hit up hypothalamus to release more ADH to posterior pituitary gland, and that would travel by blood and go to the collecting duct membrane and cause more aquaporin to be inserted in there. Okay. I don't know if I drew a picture. I didn't draw a picture, but basically, if you have more aquaporin, more water would be reabsorbed, right? Great. Similar thing if you have increase in osmolarity, that is detected directly by hypothalamus osmoreceptors to trigger this pathway. And if you have more water being reabsorbed, blood volume would increase, blood pressure would increase. Okay. I think I've said that before, like whenever you're answering a question, like do follow this pathway, like say that, okay, this increases, so that happens, is detected by that, blah, blah, blah. And in the end, you have increasing blood volume, increasing blood pressure, like follow the whole thing. Another thing, a quick pro tip, while like if you feel overwhelmed about like too many questions in the exam, don't have enough time. I don't know how you feel. Do you feel like there's like less time to answer all the questions, or how do you feel about time? Yes, no. Okay. Okay. Because you do have to like write quite a bit of mechanisms. If you feel like you don't have enough time to write in like full sentences, just like draw these and like try to write in like, okay, this increases, so that decreases and stuff. You are going to get the points if you write the mechanisms correctly, but do write the mechanisms, right? Okay. All right. So that's, oh, I drew it here, aquaporin. Okay, whatever. All right, so that's kind of like ending the process of like, I don't know, filtration, excretion, I guess. Okay. Let's talk a little bit about, and I know I've posted a video on this already, but I'm gonna go very quickly over this because this is like very, very important. And I don't think the lecture slides has it like that many slides dedicated to like the whole picture. But you are gonna get a lot of questions on this. So just wanted to go over it again. So what happens if you have low blood pressure, right? So if you have low blood pressure, that means you'll have low GFR. That kind of makes sense, right? Okay, so your body would want to, remember homeostasis, they want to bring everything back to normal. So their goal would be to increase blood pressure. Their goal would be to increase GFR because they would want to like maintain those things, right? So how do we do that? Let's see. So we talked about the whole process of filtration that happens in the glomerulus, right? Okay, so if you have low GFR, basically think about it, you're going to get like low volume of filtrate into the lumen, right? Less sodium chloride and everything. That could be detected by macula densa, which is sitting in the ascending or descending? No, wait, descending, right? Yeah, descending. Okay, descending loop of Henle. So they would be able to, because they're like right beside the glomerulus anatomically, and they will be able to detect that fall in osmolarity. And they will send, generally they send like paracrine signals to granular cells, which are sitting right in the lining of afferent arteriole. because of detecting low osmolarity, they're gonna say low signals to granular cells. That kind of triggers a few things. Is that okay till now? Okay. So what does granular cells do? They just sit beside the afferent arteriole, right? They would get the signal from the macula densa, and they would make the afferent arteriole more dilated. Okay, they would relax, make the arteriole, afferent arteriole more dilated. Yeah. Uh-huh. I don't think so. No. Yeah. Yeah, I don't think you have to. Basically, I think ATP, did you say ATP as like signaling molecule? Yeah. So adenosine itself is like a vasodilator. Yeah. Yeah. No. Yeah, it's not that. I thought nitric oxide was also relaxer. Yeah. You do have to. You don't have to know that. Yeah. So as long as you mentioned like what's triggering what. Okay. So yeah. So following that line, it causes the afferent arterioles to dilate, and that's the tubulo-glomerular feedback that you talked about in class. And if this dilates, that means more blood can like go into the glomerulus, if you think about it, and that means the filtration rate would be increase, decrease, higher. Yeah, exactly. So, yeah, which is like one of our goals. So like, check, awesome. Another way they do this is they just intrinsically they're able to detect the fall in pressure and then relax. That also like dilates it. So that's a myogenic response, but I don't know if you guys learned it.
The granular cells themselves.
Yeah, yeah. So arterial, afferent arterioles themselves like good. Yeah. Okay. They just like normally relax as well, so they dilate. But another crucial thing they do after getting these lower signals from macula densa is they release renin. Okay. This part is a little bit important, and I feel like nobody wrote it correctly in the I think case study questions. That's why I want to like emphasize. So the signals that cause renin to be released, there are three. One is that direct signal from macula densa. That's one. Second, you will have like a direct detection from the granular cells, right? From the afferent arteriole, like they directly detect like, hey, pressure has fallen, must release renin. Cool. That's second. Third is sympathetic nervous system. So you do have, if you recall from like previous parts, that sympathetic nervous system also has like a connection to the kidney. This is where they have it. So they can release renin if you stimulate the sympathetic nervous system. And if you also recall that if blood pressure falls, you do stimulate sympathetic nervous system as like to counter it, right? So you do have increased sympathetic nervous system, and that causes renin to be released. So these three pathways to release renin.
And it's like, where is renin actually?
Granular cells. Cool. So renin is released. Fine. What do? They go into the blood. They find angiotensin, tensinogen, which is released, which is made by the liver. I just like wrote it, but liver. And they would be able to convert it to Ang1. So angiotensinogen is like inactive. They convert to angiotensin 1, and then angiotensin 1 is converted to Ang2 by ACE, which is mostly released by the lungs. And then angiotensin 2 does a variety of things. Okay, but what is their goal? Basically, they want to increase blood pressure and increase GFR, right? So they're gonna fulfill those goals. Let's see. So one thing they do is they constrict the efferent, which is like here. which is great because if you constrict this, that means more blood is going to be backed up here in the glomerulus. GFR would increase. Okay. Then they also trigger the cardiovascular control receptor to increase blood pressure, like do all those things that they do, which is like increase heart rate, vasoconstrict. They themselves will be able to constrict the arterioles to like increase TPR or increase blood pressure, right? So these are all like cardiovascular stuff that they do. Cool. Another thing that they would do is they would also trigger the hypothalamus to increase release of ADH. And if you have more ADH or more vasopressin, you're gonna reabsorb more water, blood volume increase, blood pressure increase. Okay. Always do write whenever you write like more water is reabsorbed, more sodium is reabsorbed. Do write immediately if your goal is to show how blood pressure increase. Do write blood volume increase, blood pressure increase. Do write those things. Okay. You're also increasing thirst, and that's going to increase blood volume as well because you're going to drink more water. Ang2 also causes the proximal convoluted tubule to increase the reabsorption of sodium. I don't know how much they have talked about it in detail, but they haven't. Okay. That also increases blood pressure, obviously. Another crucial thing that you did talk about quite in detail is Ang2 causes the release of aldosterone from where? Yeah. What is released from adrenal medulla? If I say like blood pressure is low, something is being released from the adrenal medulla, would you write aldosterone? No, right? So that's a trick question. What would you write if I say something that's being released by adrenal medulla has increased? Yeah, using. Yeah, epinephrine, exactly. Right? Okay. Let's not confuse those two. I confuse those two all the time. All right. So aldosterone, what does it do? It increases sodium reabsorption in the distal, and we talked about it briefly, right? It incorporates the ENaC channels so that more sodium would be reabsorbed, blood volume increase, blood pressure increase. Is that okay? Yes, no, yes. Yeah. All right. So we have kind of fulfilled our goals, which we wanted to increase blood pressure, which we did because we increased reabsorption of sodium mostly. And then we also increased the GFR because we caused the afferent to dilate and efferent to constrict. Right. All right, is that okay till now? Okay. So another final thing is when the blood pressure is increased. So we talked about what happens when blood pressure decrease. In the blood pressure increase part, everything is just the opposite. And there's one additional thing, and that additional thing is ANP. Okay. And you guys talked about it in class, but just like think about what it does is when you have higher blood pressure, the myocardial cells would release ANP, just atrial natriuretic peptide, and that would cause a bunch of things. It would cause more like less ADH because again, you don't want water reabsorption because you want the blood volume to fall, right? And then they would also decrease sodium reabsorption in the distal convoluted tubule so that less water is reabsorbed. Less sodium is reabsorbed, less water is reabsorbed, blood volume decreases, blood pressure decreases, all the stuff. Okay. And they would also like reduce sympathetic nervous system, reduce aldosterone, basically anything that would get to their goal, which is they would want to reduce blood volume and blood pressure. Okay. This part could be a little bit confusing. So they also slightly dilate the efferent just because they want to get like more blood to go into the glomerulus to filter more sodium. So that increases the GFR slightly, which is not your goal, but yeah. Is that okay? Yeah.
So
Hmm.
sorry, so we want to filter more.
Yeah, but that's like slightly. So that's like partial. I'm just gonna like do bracket, to be honest. It's like not even.
Less in the blood.
Yeah.
Okay.
So that's just generally you would not want to do that because you would want GFR to probably reduce because the like blood pressure has gone up, GFR has gone up. But this is just like counterintuitive a little bit. Yeah. Is that okay? All right. Okay. Which one? This part? Yeah, so this is just a little bit counterintuitive that they would want to decrease GFR, right? That's their goal because in this case, blood pressure has increased, GFR has increased. So you would want to decrease it, but ANP actually dilates the afferent a little bit, just because that helps them get more blood in so that more salt could be filtered because they would want to filter more salt at the end, like that's their end goal. So yeah. Okay. All right. Yes. Yeah. They would always go in hand in hand because the thing is to realize is blood volume is not something they would want to like keep constant, but like that's not technically that's not the goal. Indirectly that does happen, but the goal is always like osmolarity is kept constant. Yeah. Priority is osmolarity. So whenever, let's say, for example, you eat a bag of chips, right? They would want to keep the osmolarity constant. So they would actually reabsorb more water. So your blood volume would increase, blood pressure would increase slightly, but your osmolarity would be kept constant.